The overall goal of this procedure is to use a streamlined method to purify plasmid DNA from E. coli. This is accomplished by first growing overnight cultures in a deep well plate. Then, harvest and lyse the bacterial cells. Next, following precipitation of protein and genomic DNA, clarify the cell lysates. Finally, isolate plasmid DNA on the DNA binding plate. Ultimately, this system offers a higher yield of sequence quality DNA using a cost-effective procedure.
The primary advantage of this technique over prepackaged kits is this method produces greater amounts of high-quality plasmid DNA at a significant cost savings.
Using deep well plates, inoculate 1 milliliter per well of luria broth containing appropriate antibiotic with E. coli transformed with the desired plasmid DNA. Culture overnight at 37 degrees Celsius. Pellet the E. coli in the culture plate at 5,000 times gravity for 10 minutes. Then, decant or aspirate the supernatant. Resuspend each pellet in 100 microliters of resuspension buffer containing RNase A. Add 100 microliters of lysis buffer per well and mix using a plate shaker for 2 minutes. Add 100 microliters of neutralization buffer per well and shake for 2 minutes.